I want to talk about how this novel coronavirus was identified and sequenced and how that led to new diagnostics. Well, there's a number of ways which these can be investigated, and we've had experience of doing this many times as scientists. I was involved with the SARS coronavirus doing this. But in Wuhan, China, what they did is that they took clinical material from the patients. It's a lung wash is what they used to start this. That material was put into the tissue culture to see whether you could identify the virus. The same material was also used in standard molecular assays or multiplex PCR type assays, and this allows us to look for all the common types of viruses. But also we look for generic ones, so we included a set of primers which could detect coronaviruses in general. And finally, some of the material was put into the deep sequencing. So this is the new generation of sequencing, which allows you to do very large sequencing jobs and look unbiasedly at the material and see what can be discovered in that process. All of these approaches gave the same answer, which is a little bit unusual as a scientist actually, but they all gave the result as a novel coronavirus that came out from this. Even the electron microscopy that looked at the virus looked a little bit different from the average. Tissue culture also gave slightly different effects from what was expected. The PCR multiplex molecular assay also gave a generic coronavirus and none of the specific ones. And then the deep sequencing identified the whole genome of a coronavirus which looked somewhat similar to other bat coronaviruses, which was also distinct. And from all of this, we were able to identify that this was a novel virus for humans. So this work was all done in Wuhan, China, and also with in Beijing, the CDC group there, and effectively done in January 2020. So very soon after the first cluster of cases were done. From the genome sequence, we were able to identify the novel regions of the genome and also therefore develop new molecular diagnostic tools from that. And certainly in China, that was done within just a few days, and that was allowed the diagnostics which everyone is using. But because the genome was published, actually all the other CDCs around the world also developed their own primers. And some of those overlap, some of them are similar, but because there's a diversity of locations across the genome, in fact, there are now several different assays which can be used to confirm each other. So this was a huge aid to the rest of the world, and these diagnostic assays could be produced outside of China and around the world. Well, as I mentioned, it's a very fast process once you have the genome. Looking at the molecular assays is a matter of designing the primers and putting that into a standard type of assay, and that can be done in a few days. Of course, we're all hoping for the much simpler and easier to use point-of-care diagnostic, and I'm hoping that that might become available, but the technology for that is a little bit more challenging. We are also hoping that we might be able to get serology type of assays. That's looking at the immune response, the antibodies to the virus. And that test would also allow us to have a second analysis to allow the diagnosis, but also might give you some history of whether you've had the disease in the past. And additionally, this type of assay is the more commonly used one for point-of-care diagnostics. So there's some hope that we might be able to improve the diagnostics in time.